Okay, so we are going to do multiple alignments today, but questions? No. So this is about this slide here. Science is communication, so questions are the beginning of it. Talk to each other, use more of this opportunity, ask me. Again, the way we look at 3D structures is 3D coordinates. The same information is in 2D. In 1D you lose the information. On 1D you have the sequence, secondary structure, and it's just a string. This one, you can produce the 3D structure, from that one not. And the lecture is about sort of using the sequence to predict 3D structures. Here I show you from the Cath superfamily, this is work from Christina Renko, originally with Janet Thornton, and then they went on and do that alone now. So what you see here is a superposition. Everybody in the room know what they see? I mean, I'm not talking about the detail of the particular protein family, this is not what I'm talking about, but how this image is created. So this is a superposition of an entire family of proteins. So we have many proteins for which we have the known three-dimensional structure. So there are many structures underneath, and we know that they are similar in their 3D structure, and they're all superposed. For this particular PLP-dependent aspartase aminotransferase, or like the major domain, so it's only one domain of that. And you do see that essentially all of those have helices here, although when you superpose them they align at slightly different places. But nevertheless, the main theme here of the helices and these helices and the beta strands in the center, all of that theme is completely conserved in that family, right? And that in fact is what people then would assume is the theme that is relevant for the function of that protein. That is why it's conserved. These kinds of things we are trying to find by alignment, so by using a sequence and querying the database. The simplest one is dynamic programming, is brute force. There's a clear solution. Clear by the definition of this is the optimum. This is what we want to optimize. This is the function we want to optimize, where this essentially is the lookup sequence, or if a delta function, if we look at identity, gap open, gap elongation. So which is the method underneath that sort of shows that it works, is the famous Smith-Waterman, Temple Smith and Michael Waterman, 1981 method. And by the way, both very, very nice gentlemen, both Temple and Michael, and very different gentlemen. So one way of looking at it is the optimization of sequence identity, as in you match the letter. And the other way, okay. So if you do not have identity, then you want to match something that is more related to biophysical features, then you look, for instance, at the Blossom matrix, and you see the diagonal elements are not all alike. Matching cysteines is more important. Essentially, that's why the diagonal is here much higher, or tryptophan is a similar thing. And least important, if you want, is sort of an S here, matching an S on an S. Okay. There are different words out for what I show here, and the way to read that, you can call it a scoring matrix, a scoring metric, a scoring table, substitution, and so forth, right? So we have two words and all possible, and you will find all of these words. It's all the same thing, and I use them I use all of these here. The most frequently used one is the Blossom 65. BLAST is a fast matching algorithm that simply looks for the identity of words, then extends the words, and in between it does do a match. It works only if you settle the statistics, right statistics. This is the score that I showed you, Smith-Waterman score essentially. And that is the score distribution for one particular alignment of the background, so 111 million in the database that you have. And that is one pair of proteins. That's the hits. So these are two alignments here. And you ask, how much do the two alignments that I see differ from the background distribution? And what is important to compute the expectation value that computes this difference here is how far is the score from the average here, and how wide is the blue distribution. Okay? Those are the criteria that you need to use. And then the expectation value, essentially 10 minus 3, so you have an e-value of 10 minus 3, that means that the chance that this value that you have has arisen at chance is 1 in 1,000. Okay? Is that a high number? No, why not?
Because the database is so big.
We have 111 million. So in 111 million, 1 in 1,000, you can do the math. You will find 1,000 of those hits. Okay? Put another question to you. If I always give this example, a blast started with a word size of 3. So the simple match in the word here originally was 3. For 3, we have 20 amino acids, 20 times 20 times 20, 8,000. So finding 8,000, any word of 3 that you can make up of 20 amino acids is going to be in our database. We have 111 million sequences. Each of those at least sort of ballpark 400 residues long. Is roughly 100 million times 100 million. And so any 8,000 you will find there very, very often, right? So any word you make up is in the database. You have to understand that. And this is just sequencing mistakes essentially get you into this reality, given the extremely vast space that we're already covering with the sequence databases. So today you really need to tweak these parameters in the way you do it. You need to stay away from the background more than you used to in the past. When we do pairwise alignments and we do look at sets of proteins for which I know from 3D comparison that they have similar 3D structures in blue, and we compare it to pairs of proteins that we know have not similar 3D structures in red, then we see that the twilight zone here, there's a safe zone in sequence alignment or inferring structure from sequence alignments. There's a twilight zone and there's a midnight zone. The safe zone is where you can clearly make the inference to the twilight zone where you lose the signal. And that one here shows you the twilight zone where you see that if you're above this twilight zone, if you're in the safe zone, essentially you don't make mistakes. There's no red pair. There's no pair. At this threshold, there's no pair. This one here is 10 or close to 0. And at the same time, at this very point here, you are this is an logarithmic scale, so you're at a point here where roughly at 8, I don't know, you have 6,000, 7,000 or something like that is where here, where this is 10 and the blue is 6,000. So essentially anything, most absolutely 6,000 of the ones you find at that point are blue, meaning they have a similar structure. 10 are red, mistakes. So essentially you don't make mistakes. Okay, that's why this is called safe zone. But then you see that within 1, 2, 3, 4 points here of percentage sequence identity, we get from not making a mistake to 50%. So blue and red are alike, are crossing here, the lines are crossing. That means 50% of the ones, if you call everything that is above that line to be blue or to be related in structure, 50% of your decisions are wrong already at this point. Okay? Important to see is, so the red explodes immensely. So over 1, 2, 3, 4, 5 orders of magnitude over a course of 20% sequence identities, 20 percentage points, right? Within an interval of, so you go from 100 to 0%, and the twilight zone is defined as something that is sort of 20% long, and in this you shoot up by 6 orders of magnitudes in the false positives, in the ones that are not related. But at the same time, in the same interval, the blue ones shoot up by an order of magnitude. Okay, you may say, compared to the red, the blue is not really rising a lot, but it's an order of magnitude. So you, again, let's follow up on this one here. So you're somewhere here at 6,000 and you're crossing over to more than 12,000, again, within 5 percentage points. So from here to here, it's doubling. So the kind of things that you want to find, and here it rises even more. So the things that you want to find, the relations that you want to find, are in the twilight zone. You want to have methods that find them. The problem is the red line. Okay? So ultimately, what's the way out of this dilemma? We could try to improve statistics. It's not enough. What can we do? You have heard of that. What is a better alignment type or a better way of aligning sequences than pairwise?
Multiple alignments?
Yes, exactly. So multiple sequence alignments. So let's begin with the negative aspects of multiple alignments. There is something about problems. So the first one, if I wanted to sort of do in a dynamic programming of say 3. So pairwise I showed it all works. For 3, it is a complete problem. It gets complicated. There's some claim that you can do up to 6, but you need 60 terabytes of main memory. But 6 is small. We all know that today families typically are 1,000 proteins. Okay, so 6 is not a number that is enough to model 1,000. There are the first hack again. Getting back to David Lippmann largely, so worked in the past when the goal really was more like 6 or 10, but with hundreds and more, this no longer works. This second hack that was introduced by Russell Doolittle here does still work. And that hack simply is, you take, in this particular case, I show the example for 4 sequences, you take your sequence, you find in pairwise alignments many sequences that are similar to it. And then you call them a family because all of those are similar to A. And then you simply do an all against all. Okay, all of these are pairwise comparisons. And pairwise comparisons we know how to do. Now in pairwise comparisons then now you have essentially two steps or 3 steps, and one way of doing it is the following. You take the first pair being defined as what is most similar to A is B, and then this is you align A and B, and you sort of build a consensus sequence. In this particular example here, I made a mistake. I just, as a consensus sequence, I sort of picked wherever the letter differs, I picked the first. And in the example is wrong because then this is like not aligning them at all. If anyway I always take the first sequence, I completely throw away the information from the alignment. So you could reproduce or you could change that by simply taking the most informative residue at any symbol and any. So wherever they differ, you pick the one that has a higher value on the diagonal of blossom. Okay, this is a simple way of creating a consensus algorithm that in a statistical sense makes sense. And then you create a new sequence that in fact in this particular case would be then none that exists. Okay, so you do that in the second step. You take whatever, in this particular case only 4 examples, so it's only 1 pair left. You align this 1 pair, you create a consensus sequence, and then in the third step you simply align the 2 consensus sequences. This is the way to think about this. This is not necessarily a way to do it because this idea of consensus sequence is really putting the sequence out there, and thereby you can match it to dynamic programming. We see now back to the same problem, right? In reality, and we will do it in sort of a probabilistic way, meaning we will replace the sequence by a probability of it being there. But I only illustrate the idea of the hack. Now this is one way of doing it. Here's another way of doing it. In the first step we do the same thing, but in the second step, rather than sort of looking at the second pair, you take the consensus sequence and align it against the third. And then in the third step you would take the fourth and align it against the others. So essentially, I'm not sure where I got the zones back. This, in fact, now means that you sort of build up something that is referred to as a profile, PSSM, or motif. And a motif underneath, so the slides are from Teresa here, the motif idea here is illustrated by her on the transcription factor binding side. So this is not a protein sequence, this is a genome sequence. And simply the idea is an information content. The height of the letter is proportional to the information that is gained by that letter, meaning you immediately see that there is a motif here for CTG. So the probability of CTG, the CTGs that you see at the positions -7, -6, -5 is much higher than expected by chance. While, for instance, at position 1, or position 1 is good enough, or position 2 here, at position 2 there's really almost no letter none of the 4 letters really stands out. So they're all sort of equally possible. There's nothing special to be said about position 2, which is a statement too. So the statement is -5 here is important, and not only is it important, -5 and +7 are the most important positions, so the highest. And in 1, -5 is a T, and in the +7 it's an A. Okay? And there you immediately know what sort of defines your, in this particular case, transcription factor binding site. And now, this is essentially like a motif, like a regular expression here, and the matching sequences to this regular expression would be, for instance, these. Okay? And by looking at this, sort of, you could perceive this as a multiple alignment also. By looking at this, you sort of could extract again from a multiple sequence alignment, you could extract the motif, or you could simply look at the PSSM. Building the PSSM, you sort of begin with absolute frequency, turn them into pseudo counts, relative frequencies, and log likelihoods. This is all math. Ultimately, you sort of normalize by what the background probability is of these, and then you essentially get to the log odds, and the log odds, now that tells you at every single of these positions, 1, 2, 3, 4, 5, 6, 7, what's the probability of having one of these 4 letters in there. And then sort of more generically, we have that in the Blossom matrix in some sense, but the difference between the Blossom matrix and this way of doing it is that the Blossom matrix shows you what's the probability of having a Q exchanged against a D anywhere, whether it's the first Q in the sequence or the 5th Q, makes no difference. Or let's, in the next example, I'm going to have a V. So the V on the anywhere in the sequence doesn't make a difference. What I show here is 4 different sequences aligned. For the first one, for instance, there's one that has a V, and the V on the alanine here has a 3, while at position 9, the V on the alanine has a 5. So once we have the counts, so these are created by, you take pairwise alignments, you find 4 that in the database that belong together. These 4 are aligned here. Once you have the 4, you build up a position-specific matrix. The position-specific matrix, again, the 0 approach without any count is you take Blossom, or you take any background matrix. The moment you have the first count, you change the Blossom matrix, or you change the matrix to a position-specific matrix, but in this particular case, you see for the V at position 1 and the V at position 9 gives you a different column. Here. The values in that count are different. So whether you align the next sequence coming with a V at position 1 or position 9 will be differently weighted, okay? With whatever residue you come in the next sequence, whether it's an alanine as the example that I showed, or even the match of the V, I guess. Let's see whether I'm right here. Yes, the match of the V here is 10 at position 9, and it's only 6 at position 1. So whether you want to align at position 1, you care much less to align another V than at position 9. Okay? Is that clear? This is, I like to dwell on this. Is there anybody in the, I'd just like to pause on it, because this is a very crucial concept for the remainder of the lecture. Is the difference between a Blossom and position-specific matrix clear to everybody in the room? Please stop, please ask some question. I also wanted water. Yes, yeah, thanks.
So at a certain position, the V appears more times, then it raises the number. That's basically.
That's essentially exactly what it is. So sometimes, let's pick an example here over which you may stumble here. At position 7, 2 of these sequences that we used have an S. One has a Q and one has an E. The consensus sequence happens to be an E. I do not know how they computed the consensus sequence. But what I do know is that the SS I showed you in the Blossom matrix before, the diagonal of SS is very low. So matching an S is not as important as matching a polar or negatively charged residue, or two polar residues here. So now ultimately, so I have a 50-50 here on something that is SS sort of free of a clear feature, and something that is polar charged, right? So polar charged, there's only two two negatively charged residues, only a few negative polar residues. So the random odds of showing that if you have two in 4 already there that have some feature, then maybe feature is relevant. And once you are in feature, then you have to essentially pick between the Q and the E, and you pick the E because that again has the highest value in the diagonal. So it's not always the number that is highest, but it's the one that is most surprising in that sense. And again, if at that position you would add another E, you would substantially add to that. You would add another S. At some point, this would become an S conserved is important. So at some point, that would flip. And at some point, any position, so the most extreme one is the alanine. So when we see that an alanine is conserved, there are very few conserved alanines, and those are extremely important to match. Or glycines. So glycine and alanine are typically easily to replace all other residues by. However, bless you, once you see conservation of these, then they give you very, very strong signals. And again, the way it happens is by just adding more and more sequences to this alignment, you build up this matrix. Yes?
What does the plus and minus in the last column mean?
I do not know. Bless you. I do not know. Sorry. Oh, maybe could it be the sum of the plus? No, it cannot be. No, I don't know. Yeah, I suspect that this is the difference between the negatives and positives. No, I don't know. I'm back to my first explanation. I don't know. Sorry for there are two things I don't know. This one I didn't remember that I don't know. I did not know that I don't know. I clearly don't know how exactly they computed the consensus sequence. But let me take this as a moment to also hold. The consensus sequence has no reality. It is something that I use in order to explain the hack of Doolittle for multiple alignment. It's something that is sort of intuitive, but methods, very few methods really use consensus sequences these days. Underneath what you use is a probability. These numbers here, that's essentially the number that you use for alignments. And ultimately, profiles profit from simple signals, such as there is always something aligned here, there's always something aligned here, and the gap, if you put a gap, then put it in between. And that brings us up, brings us to the other question. What's the difference between a profile and a motif? Regular expression motif, for instance. Yeah?
I think a profile is a whole sequence of the protein, and the motif is only one.
That's it's a very, very reasonable definition. I believe I would have tended to give exactly that explanation for most of my life. Today I would rather sort of see it as something that tends to be motifs. We tend to look more, it's true. So for most situations a motif is a shorter thing. Another way of looking at it, a profile tends to rather reflect a probability model underneath, which again is sort of also in the sense for an entire sequence or an entire domain alignment. But it's typically size and probability versus regular expressions tend to be the example, tend to be the background. Here's the psi blast. The psi blast essentially is exactly the idea of beginning somewhere with this generic blossom type of matrix and turning it into position specific, and that's the Pierce of the Psi4 matrix. It again goes back to Stephen Altschul, who is behind this, and it's just one of those examples. This is a few years ago, but in some sense you may argue, so there are many people in the field, and some of these are even, at least one of these is in the room even, who get higher H indices. But nevertheless, so the influence on bioinformatics ultimately is incredibly different from Stephen Altschul. It is all in changing something that completely, so the H index is not always measuring the influence on a field. Stephen Altschul clearly has influenced the field, I believe, more than most of us. Another important person behind is David Lippmann. I already mentioned him today a couple of times. Again, with mostly he's the director of the NCBI, has been the director for the NCBI for a long time. But again, he's also on all of these important alignment publications from the early days. And that gets to another person on the early PsyBlast, is Webb Miller, Tom Madden, and Alejandro Schaefer. So over 50,000 citations. What's the guess of this, the sketch of the solution? Fast hashing, we already talked about that, the word match. Then after we have the word match, we essentially do an extension that is, by the way, the word match is no longer a word match in the PsyBlast. It's essentially a word match in the sense of the Blossom matrix. So it's not an identity word, it's a Blossom word, and getting that right is highly non-trivial in terms of statistics. Then there's dynamic programming extension. That's another big difference to the original BLAST. So whenever this hit here is extended, it is done by dynamic programming. I presented that in the last lecture as if BLAST did that, but it was actually only introduced with this publication in '97. Originally BLAST did not do the extension that way. It did it in a very roundabout, adding if the matrix is below or above some value, so not really in dynamic programming. But ultimately we discussed that already. But it technically came, why is it so fast? So why can we speed up over dynamic programming so much with PsyBlast, although we go back to dynamic programming? And the answer to that was because the dynamic programming is the square of the length of the two, and essentially by extending here, I extend over something that's a relatively short segment. So I'm not doing dynamic programming on the full length of the two. I'm only doing dynamic programming on something that is small. So if it's only half, you go down by a factor of 4. If it's a fifth, by 25. This is 1/5 by 1/5. And that's the factor that you gain, and that by shortening, by short matches, you speed up tremendously. Anyway, then comes the statistics, the expectation value, which in fact also has improved. But then comes the real new step. So you go one sweep, pairwise alignments against the database, 111 million, and you build up this profile. Okay? Now, in the next step, what are you going to do? So far, we have done nothing really new. A few minor improvements in the 97 paper over the 93 paper, but nothing big. Well, now, the big thing, we have a position-specific matrix. That's where the Pierce in Psi comes from. But what do I do with it? I put it on the whiteboard. Something else I can do. You really don't know? What do I use? We're back to the profile. I have this profile. I asked you, do you really understand the profile? What do you do with the profile? It's simple. It really is simple, no?
Maybe if you know the previous structure of one sequence, you can infer it for
Yes, you could do that, but much simpler. I believe the way I ask the question is let me just, my question is too trivial. So I take a sequence and I line it against this profile. So in the first run against 111 million, I got a profile. And now what I do in my second round is I take every single one of the 111 million in the database and I align it against the profile. So I'm no longer asking, is this as a database sequence similar to my query? I'm asking, is this database sequence here similar to my query profile? And that's the point where the valine has at position 1 a different propensity of fitting another valine than the valine at position 7 in the example. Okay? So in this particular example here, I have sort of highlighted here 4 conserved columns. And in those 4 conserved columns, any of my 111 that I pull in has to fit more to these conserved columns, okay? And then the I in the PSI blast or Psi blast is the iteration, meaning that I do that again and again and again. So every single time, so I do 111 million database sequence against a profile, and say in the first round here I found 50 through my pairwise alignments, and the second I add another 20, and then I change my profile. And again, simply by counting at every single position. So I add more valines, I add more E's in there, the profile is going to change. In my second iteration, I'm running against the change profile. And I keep going. What's the termination condition?
When the profile stops changing.
That is what we have always said. In reality, we cut out after a fixed number of steps because there was not always enough CPU time around, or because of something else that I get to in a moment. But in principle, yes, when there's no more change. That's the simple convergence. Now, and let me show you somehow the idea of what PSI-BLAST or what any kind of profile search does. It's just that PSI-BLAST made it possible to do this sort of thing in terms of CPU affordable way. So say this is my family, and this is my family defined as everything that I know for a particular protein family that has similar 3D structure. I don't know it today, but maybe for some family I have a large family, and say the family would look like that, green. That's what I want to get. U is my unknown protein. I want to map U to other proteins. Okay, if I do what I showed you before, remember at some point in the pairwise comparison I had this red curve where proteins have different structures, and blue where they have the same 3D structure. I showed you today how in the twilight zone the blues explode but the red explodes more, and by that I simply can build a circle around U. So I find everything that is safe at the distance to U. So above this, wherever the red, remember on that slide, wherever the red was 0. Or close to, it was 10 to 5,000. I call that 0. And that's the threshold that I built. And by building a threshold, essentially I define a circle. I have my sequence U with which I search, unknown protein, and I find everything within, it's a circle because it's completely symmetric. I have pairwise comparisons, right? Everything that is equally away from this central U, okay? Now, that's in my first round, my safe zone. Now in my second round I build a profile, and this profile will allow me to look into this lens here. This lens means it's a little bit larger because the profile allows me to extend more, and it goes more skewed in a particular direction, and the particular direction it is skewed in has to do with where were the sequences found. So in this example that I show, there's U and 1, 2, 3, 4, 5 others. And they happen to be sort of not completely centered in that circle. They happen to be more on this side. It could be on the other side, it could be wherever. In my example, they are sort of here. And because they are grouping more on this size here, the lens is around them. Because the lens is defined by whatever I found in the first round. Right? As you said, whenever I add another V there, I change my profile, and I can only change it according to what I see, my 6 sequences, the U and the 5 others. And again, so this allows me to reach a larger view. What you have to remember in your PSI-BLAST in the last readout of PsyBlast, you have those. Because you didn't find anything here. But there may be some lost. You may have had one in here that in a higher run in PsyBlast are in the safe zone. In a lower run where things get better, this simple initial finding is no longer there. And that comes because the lens drifts slightly away. And what used to be safe is no longer safe. But you should sort of take that back out. Because whatever you have on a higher order run, so if this lens here is iteration number 3, whatever was there before, you trust clearly to have been right. So that is one thing that you should not forget in using PsyBlast. That sometimes information gets lost. Okay, so the next issue is, and I did not get into that, but so I'm not going to get into it. Full stop. Now, there is another issue here. If by mistake, at some point, you bring in something from a different family. Maybe until that point, before the red came here, until this lens here, you could have had 100 pairs already aligned. And one from a different family can change the profile dramatically. Why? Because remember, in this profile you build up that at one particular position you have to be absolutely conserved. If you pull in the wrong sequence, by whatever reason makes you pull that out, this, what used to have been conserved, suddenly becomes not conserved anymore. So you change a very important, crucial feature into something that is no longer crucial. So you had to conserve that residue, and this one suggests you no longer have. How does it then happen? Well, those could be borderline cases. Those could be cases that sort of everywhere else for the non-conserved position, they add a little bit of a signal. And the signal could just add up such that you always have to have some threshold. All right? That by that threshold, maybe this just jumps in. And then you recompile the profile with this one, and then what will happen is this one will open the lens for this family, that in this example here is chosen such that it's unrelated, so it's a different structure. And the moment you begin to do that in the next, so first of all, the iteration will no longer converge. And secondly, in the next round, it will drift more and more into a profile that is sort of meaningless, essentially. And that's the danger. How can we avoid that danger? Well, historically, what people have done is that's another way to stop. Well, one criterion for stopping was you wait until convergence. Another idea was you simply stop after 5 times. And another idea was whenever, and that goes back to David Jones, who I have heard talk about it first at least, now at UCL. David Jones sort of simply asked, well, if in my one iteration plus, so I plus 1, suddenly I double the number of sequences I find, then I'm suspicious that I'm pulling in something that is totally wrong. So when between I and I plus 1, the number of sequences added became too large, he stopped. He said he did not accept the last iteration anymore because he was afraid that then there was pollution in fact, that is a hack that many of us, including we, implemented at that very point. So if one next iteration brings too much. Essentially, this is the idea of PsyBlast. Cluster W, essentially, the cluster W, cluster X, have you guys heard of it? Essentially does a profile-based alignment using dynamic programming. This whole series of programs, cluster Omega is the last, no, it may not be the last one. It's 2011 now. Going back to the idea of pairwise alignments, going back to the Rost doodle approach, difference between Clustal and the hack is that Clustal builds a nice phylogenetic tree and then in fact uses varying substitution matrices. So they align any pair of sequences at any point, given the similarity that they have, they choose the substitution matrix. So within one family, they will use many, many different matrices, substitution matrices, depending on whatever they see. That's one thing. And then they will build up a tree. One issue about Clustal is time. So this Clustal omega shows here that it sort of scales in a way in which Clustal is in red here, Clustal omega. The time is somehow still okay in terms of number of sequences, a number of families, members of a family here. So it is sort of doable until numbers, even in the 10,000, they say at some point it gets more complicated. Today we are sort of reaching these. So there are some families that reach 100,000. There are methods out there that need more than families that are larger than this. And that's sort of getting to the point where Clustal is no longer working. But Clustal is an outstandingly known method, over 100,000 citations. It goes back to Des Higgins here. As another point, H-index not very high, but for a long time was the highest quoted person. Toby Gibson, so Des is essentially the one who understands dynamic programming. So from him comes the entire dynamic programming solution of Clustal. From Toby comes the intuition. So Toby Gibson is the person who really can visually align sequences and tell you, looking at it, tell you what is right and what is wrong. And from Julie Thompson comes the part that is the visualization. And ultimately I'd argue that the reason why one of the most important reasons why Clustal is so successful is because of these three people having worked together in the right way. And the second reason I believe really is Julie's visualization. So Clustal allows you to have a nice image. And people quote, everybody wants nice images. In fact, this is something that you all should think about more, how an image could be nice in your thesis, in your presentation. Use nice images. We all like images. Spend time on it. And in the publication that you have, think about maybe your publication should be on the front line of the journal, or on the front page of the journal. Think about images that are like that. Begin thinking about it. Sort of think about your publication, your thesis, or your presentation in that sense. That there's this one thing that if anybody is going to make a movie of it or whatever, there will be one image, and that is the image that you have. Think about this. For Clustal, I claim that this is one way in which Clustal got to success. I'm saying success because there are many, many, many other people who tried to do something like that. Clustal came through. Over time, and I believe it's just because of the great team. There's another method here, Maxom, that has some historical reasons, come from Reinhard Schneider and Chrysander. And the major difference between Maxom and most other methods, including Clustal, HMMs, and T-Coffee that I will come to in a moment, is really that, well, PsyBlast is actually more on this side than on that side. It's protein-centric. So meaning that the first sequence here by query is something that is different for me because I want to build up a family with respect to this first sequence. I have my protein that I care for. Whatever, I work on it, or I want to predict structure for it, or whatever. It has some relevance for me, that particular sequence. And I want to see the universe of all families, all proteins related to it with respect to it. My sequence, my query, does not have insertions. It's just amino acids. It's a full-length sequence. And the family has insertions with respect to my sequence, okay? This is sort of a thinking about it. When you want to predict structure, this is the way you want to do it. You want to predict structure for one particular protein, not for a family. Family doesn't have a structure. One particular protein has a structure, right? And that means that everything is subdued to that. While in Clustal, the family-centric view, there is no single protein that is more important than any other. It tries to find the alignment that is optimal for the family. That's what family-centric means. And this is really a very important point. And so this goes into HMMs, which means it goes to PFAM, and this is in fact where most people today get their alignments from, because they are readily available. But ultimately, this is sort of the thing that we actually should have. And that brings us to this issue of HMMs. Hidden Markov model is simply there's a hidden state and there's an observed state. In the observed state, in terms of letters here, we essentially would ask what's the probability of having one particular amino acid follow another amino acid. And that is the transition probability. And at any point in this hidden Markov model, the decision is only dependent, what is next is only dependent on what was the state before. So a memory of only one. And by the way, many of the slides that follow rapidly now come from Marco Punta, who is, I have to update this slide, who's now in London. In the cancer center in London, and who loves coconuts. Anyway, so idea of Marco also likes football, and this is happens to be the club of Augsburg. Marco is Italian, and the slide comes from him. I don't actually know why it's Augsburg. Maybe because it's a Latin, a Roman town. Anyway, so you have some scores, and you want to predict. You want to have a model that predicts how Augsburg is going to fare in the next game. So predict essentially how Augsburg, in a particular pairing with any other team, will play. And so here you look at the lose, win, and draw situations, and you essentially, from whatever you do, you want to build a probabilistic model that allows you to compute what will be the next game. I observed something today, I observed something yesterday, what will be my next, what will Augsburg play next? And our model, for instance, there are these three states, and they are connected by probabilities. They sum, of course, to 1. They are all probabilities between 0 and 1, it's sum to 1. And now we want to sort of see one feature here that may relate to winning, so that may help us. So let's just see, when we see that they play at home, maybe they win more often. When Augsburg plays home, is more likely to win. Okay, so there are essentially two variables here, two free things. It could play at home, it could play somewhere else. And we sort of change, according to whether they play home or not, we change the transition probability. And essentially this is one way of doing it. And in this particular case here, the hidden Markov model would in fact, as you can quickly see here, relatively well model what will come out. So this will clearly be better than chance. It's a very, very simple way to look at this information. Now for sequences aligned in a protein, again, it's the same formalism. We have the transition probability, we have a state probability. It simulates a system. A system could be, here's another example, the red, yellow, and green, traffic light, right? This is deterministic. So this is not a probabilistic model, right? You know exactly what will come when you sit in front of a red light for whatever time it takes you in your, whatever awareness you have on that day, will determine how long you have to sit in front of the traffic light. But then you will immediately figure out this is deterministic. Well, maybe on some days you don't. But this example here is slightly different. So this is in fact, the weather forecast is a probabilistic, depends again on where you are. In a place such as this one here, this is probabilistic. If you go to the desert, this is not so probabilistic. You know from what you see today what will be there tomorrow and the day and the next year, the whole next years, the next 10 years or whatever. Okay, so there's a transition probability from one weather to another, and there's essentially you will try to compute the probabilistic transition from what you know today. The methods that are available there are two sort of major methods, SAM and Hammer. Hammer goes back to Sean Eddy. SAM goes back to, in fact, something Anna Schroch introduced with David Hausler a while ago, and Kevin Kapler's helped importantly to create SAM. So essentially there are two different families of hidden Markov-based models. Hammer is the one, now the latest implementation is Jackhammer, that is used to create PFAM. And again, so you start first residue, or family-centric, so some residue. And then you have essentially three ways to go. You go in insertion, deletion, or match state. And those are, in fact, the transition probabilities. Here's the way, that's sort of a generic one. This is essentially the same thing with a slightly different path through the model, but essentially the same idea, the one from SAM. And the way you define these transitions is, so you look at an entropy, and before any amino acid is there, we simply have the background probability, right? This background probability is ultimately the Blossom matrix. Okay, that's the zero probability, 0 point. Now, once you have something aligned, essentially this is like before, once you have valine added to that, you change, you have the priors, posterior probability, so what you see, and that gives you a new value. So if it is conserved, you have an entropy of 0, and if it is not, you have total variation. So conserved places here are very high letters. That essentially means that they are most surprising or bits saved essentially by the alignment, while the ones that are most indecided is where you have the 20 amino acid all squeezed together at the lowest possible information somehow, where all 20 appear in the lowest number, right? That is where the least conserved, where the least signals are, and the most important signals is simply the letters that stand out here. Okay. We zip over the hidden Markov model. It seems there's something that it doesn't like. Let me see whether this slide at least goes. So in order to build up this, we begin with a single sequence. We build a model with from the sequence or alignment, so essentially we could run a PsyBlast here. This uses the model to search additional homologs. Ultimately, in this particular case, for the first round, you build a profile. Then in PsyBlast, you use the profile and you find new sequences, and that's essentially the same in the link of HMM here. You change, one thing that I didn't say before, once you have the profile, you could also change the way you do the alignment in PsyBlast. Right? Because when I explained the profile, I said you refine the profile and find new sequences. And I did not say that you could also change the way the alignment is done, because essentially that sort of falls out of the last profile. But this is here implicit because it gets a new model, and here in the HM, this is important that you change that. In PsyBlast, the most important thing is really the last profile, and with the last profile you can restart the PsyBlast and you will get a new alignment. While here, in this iteration and redoing the alignment is in fact where the HMM gets back changed. Okay, these are Hammer and SAM. Then there's T-Coffee. This is slower. T-Coffee, essentially dynamic programming is most accurate in some sense. PsyBlast is fast. SAM is maybe even better than this one here by now. Is getting the better, the larger the family. But none of those, all of these methods here assume that there's an independence between position I and J, where J is anything else, in particular is I plus 1. So they assume an independence of the position I and I plus 1. So a residue and it's next, or okay, I plus 2, not I plus 1 in the Markov model. The next state is in the system, but not 2 states away. So these 2 residues here that are separated by 2 others are assumed to be statistically independent. Is that right? I mean, the way I looked at these, well, actually, maybe I should choose a better example. The problem is if you choose examples from real cases, then they don't look good. So I believe ideally I should have used a real sort bridge. So where it's positive here, negative here, and positive here, or something like that. Then you would immediately suspect that actually there's a salt bridge formation. And the salt bridge formation means they are in contact. And if they are in contact in 3D, they're absolutely not statistically independent. They're completely dependent on each other because they're binding, right? So this assumption is completely wrong because proteins, residues bind to each other. Nevertheless, although this assumption is biologically wrong, alignment methods work, okay? This is what, nobody does alignments in any way in which this problem is solved. There is one example that does it, and that is the tea coffee that uses a genetic algorithm, because that does not make the independence assumption. It operates on segments. It does not operate on one residue at a time. It looks at entire regions. For instance, genetic algorithm has been optimized to sort of make this spacecraft antenna here. That's $0.25. It's about that size, to give you an idea about the size. Maybe you believe that you could have come up with this concept for this antenna. But NASA spent a lot of intelligence of optimizing it, and that sort of seems to be the optimal solution, and the way that they found it is through genetic algorithm. That's one example. So the way it works is you sort of start by initialization of population. The population is just examples. In this particular case, for alignment, this would be sequences. You compute for all the sequences that you have. In our case, we don't have sequence, we actually have pairs of alignments. So we compute the fitness of the overlap of two sequences. And then, you know, if the fitness is good enough, you stop. Otherwise, you get into all kinds of changes. You roulette the parents, you cross over children, you mutate children, you compute the fitness again and go into an iteration. So whenever an alignment is not fit enough, you somehow change, you flip some residues, or you flip some regions. And then you have an alignment in the end. So here, crossover, taking from children, here mutations, so you randomly introduce mutations. And again, this is not mutation in the sense in which evolution does it, but it's the same idea. So you have a population of alignments, and you're trying to find the best one. The background of it comes from Cedric Notre Dame with Jaap Heringa and Des Higgins. And ultimately, Cedric did it as a doctoral student of Des to prove that Des is wrong and that he can get something that is better than cluster W. He did not quite get something that is better than cluster W, but it has features that cluster W doesn't have. And that's remarkable. By the way, if you want to do a study with Cedric, that is Cedric and his group at some time point here in Barcelona. And this is not Photoshop. So what you see there is the Mediterranean. This is a relatively small street. So you can step out of the institute. And there's one little street, and then is the beach. The CRG Barcelona. Actually, there were people from BIM, from Bioinformatics Munich, who, at least two, have been with Cedric. And they did not go because of the beach, but because of the science. Anyway, so where do you get your first population from? Cedric calls it a library, primary library. So the primary library, for instance, you could create alignment methods where one you use global pairwise alignment and one you use local. So this could be local Smith-Waterman and then global Smith-Waterman. That would be one way to create a population of possible alignments. Okay? And then you could also use multiple alignments. You could use structural information to sort of adjust some alignments. You use some specialist information, binding sites, important residues or something like that. And you grind all of this into the coffee maker. That's why it's called tea coffee. Ultimately, the idea for tea coffee is while the computer works on it, you have a coffee. What Cedric doesn't say is that it can be a very long, well, you go to the beach and then 8 hours later you come back. The computer's still not done after 8 hours, but that's a different story. Anyway, so essentially it still does sort of a sequence profile comparison. So it builds up a profile, builds up a population of different models and fits this one into one particular sequence into this. Again, it does allow you to take care of things that nobody else does by looking at contact information, structural information, and so forth. And it's a very interesting alternative to Clustal and to Hammer. There are other methods out there, and they all have their strength. In fact, the most important research that happens today, I had this slide where I showed the safe zone and the midnight zone. Most of the research today that has to do with alignment methods works on the safe zone, meaning aligning things that are absolutely trivial to align. Why do most people work on that? The answer in this particular case is not because it's easy, and it's there's a reason, a very simple reason. So what they speed up is, but they optimize the speed. And this is because the scooping up of organisms, so the large-scale deep sequencing, creates so much sequencing that you just want to throw out as fast as possible things that you already have seen. So the very simple comparisons have to be much faster. And that is why a lot of the modern research goes in that direction. Not on the kind of things that all these methods optimize to get deeper into the twilight zone. That's what all of these methods try to do. And again, some methods still go in this direction. So sequence pairwise goes into daylight zone, sequence profile goes into the twilight zone. And what else could you do? Sequence, so pairwise alignments, daylight zone, sequence profile goes into the twilight zone. What's next? Sequence profile.
Profile.
Yeah! Thank you. Here we are. Sometimes drops hang. Profile alignment. So simply the idea now here is, instead of going with a sequence against a profile, you compare two profiles with each other, right? And now you essentially build a profile of the entire database, 111 million profiles, and you come with a new sequence, turn it into a profile, and then search against the database. Now, in principle, this is totally trivial. Devil lies in the detail. There are too many small parameters to optimize here, and that makes it slightly complicated. And it may be unwanted. It may be that, in fact, the kind of conservations that you have within families is glossed over because you're looking too far. You're looking at relations that, in fact, would not really be the kind of relation about this pair here of residues that are somehow maybe correlated, but you would look at aspects of the family that are more coarse-grained. And maybe coarse-grained is not for your particular problem what you need. There's one cheap hack. For instance, Dariusz Przybylski, who is now abroad, did that. So for him, the way he thought about it, sort of profile sequence, going to profile, you increase the quality, but unfortunately, that's the fortunate part, that's what you want to go for, but the unfortunate part is you also increase the CPU. So can we not sort of change this here by getting a poor man's version? In a poor man's version, what I would take is get some of the quality without paying for CPU. And the way he proposed to do that is, instead of running profile, he took a profile, turned it into consensus sequence, and then ran against a consensus sequence. So this consensus sequence in that sense is like a a profile cast into a sequence, and that you align against a profile, and you can do that on the speed of PsyBlast. Ultimately, that's what he did. And then he could show that he in fact did way better than PsyBlast at the same speed, provided you have your database in the consensus sequence. So there was some CPU that went into that part. But then it got much better. In principle, that's an idea that then was extended in HH Blitz, and that's a method that comes out of Munich. In fact, that's the first one. Well, it started in Tübingen here. Johannes, who's now in Göttingen, at the Max Planck Institute in Göttingen. HH Blitz, ultimately, it comes from developments he did with Henry Lupas in Tübingen. Ultimately, you have a query sequence, you build a query HMM out of it, and then you have a database of HMMs, and you run against a database of HMMs. The trick is that he creates a pseudo-alphabet. This is like the consensus sequence, except for HH Blitz does it on a level where the alphabet is extended. So a consensus sequence now is no longer 20 amino acids. It's 1 out of 230 states. So it's something more complex, and it's not a probability vector either. Okay, it's not really a probability number. It's still essentially an alphabet, meaning that you can still do the typical PsyBlast, so you can speed it up. That's ultimately the idea behind HH Blitz, and that's one of the fastest versions for doing profile, and begins to be sort of the mostly used for that in that context. And I believe this is, in some sense, is the most successful profile version, and that does go into the midnight zone. And that I briefly get into the midnight zone. So how big is the midnight zone? Let's turn this around and let's ask for numbers. So what I want to know is at every level of sequence identity, how many proteins do I expect to have there? So I look essentially at all proteins, and I ask for all the proteins that have similar 3D structures. How many do I find at 100% sequence, or between 100 and 90? How many do I find between 90 and 80, and so forth? Here's 50. So how do you expect this curve to look? In particular, my question is, are we going to see some drop? Are we going to see some increase? Are we going to see some Gaussian? What do you believe? Where's the Gaussian? So the way we will do it is I will simply look at the entire PDB. I have to do a redundancy reduced version of PDB, sequence unique. So sequence unique means there's no pair in here that has more than 20% sequence identity. And then I compare this one to that one. Okay? Again, my criterion for they are the same structure is by structural superposition. I'm not going to look at sequence alignment. Sequence alignment is only used here to create the sequence unique subset. So this is like 5,000 running against 100,000. Okay? In terms of numbers today, PDB is about 120,000. Maybe it's 10,000. And maybe 10 against 120,000. So then the question is, how will this look? So the reason why I say there's something that starts high is because about 50% of the PDB is 100%. So 50% of the structures in the PDB are exactly the same sequence as some other PDB entry. Why? I told you that a structure costs about $100,000. Why would you do exactly the same thing for another 100,000? Because you can, so sometimes the answer is there's some truth to that, but there's also some biological relevance here. Important. So the major point is some important biological relevance. Any suggestion? Well, you know, you want to see how the drug binds. So you want to see the protein with a drug bound and the protein not with a drug bound, or the protein with another drug bound, because you want to actually see how two different drugs bind. So you have the same protein, 3 structures, same sequence 3 times, it's just different binding. And that is very important to know. How will the binding affect the structure? How will the binding change it, right? So this is why there will be a lot here. If you ignore that, then looking at very similar sequences, human and mouse is not that similar, but maybe mouse and rat is actually also not that different. But anyway, so looking at very similar things, just in order to see how, so looking at the mouse proteins is relevant because we want to see when we develop the drug, when drugs are developed, typically they're tested on mice. So it's important to see whether the protein with mouse or the drug with mouse is somehow similar to the one with the human. Okay? So there is a high probability that we find some high pairs here. Anyway, so one way of thinking about that may be this, where you sort of have either this curve or this curve. You have something that sort of looks like a pseudo-normal, local pseudo-normal distribution, and then here the gray is the background, is if you had random hits. So black would be pairs of proteins that have a similar structure, and gray unrelated things. 5% sequence identity, so this peak will be at 5 here, roughly around 5. Something to be said about that. Okay, so this is maybe somehow what you expect. And then, you know, at some point I showed this curve here. This is what you observe. And that's random. And then I told people this is actually wrong. Because when I showed them the right data first, so they never had an expectation what they were about to see. And when they saw the real data, they always said, yeah, that's what I expected. So then I began showing them wrong data. And then they also said, this is exactly what I expected. The real data looks like this. And the bar here means that whatever is on that side of the bar is in terms of height not comparable. This should be much higher. The scales are such that here, essentially I have to do an all against all. Here I do redundancy reduced against all. So the real numbers would be such that this would be much, much higher compared to this. Anyway, what you clearly see is there is no, so yes, there's this 100% peak and there is something here. But all of that pales in comparison to the real peak of the distribution. And the real peak of the distribution, as you see, is very low in terms of pairwise sequence identity. In fact, random is red, the real peak is black. You see that the real peak and random is very, very, very similar. So there are sort of 3 percentage points difference. And ultimately, these 3 percentage points difference, they make up the entire protein structure family story. Because red is random. There is no similarity between any of those. Black, similar structure. And all that they differ is sort of 3% of the residues. And those are really the important, the key residues. Anyway, what is this animation doing? These 3 to 4%, you may call them anchor residues, are most important. What it also suggests that evolution after 4 billion years had enough time in terms of structure to try all possible solutions, because we are so close to random. Maybe we can get a little bit closer. But clearly we cannot reach random, right? Because there's a difference between things that have a similar structure and those that are totally different. In terms of sequence alignment, there must be a difference, right? Because the difference is, in fact, what makes them have similar structure, full stop there. And so these two curves cannot be identical. And they're already so similar that I believe, ultimately, that sort of suggests that they're, at least in terms of structure, there is some sort of equilibrium reached. It also means that in terms of structure, really, convergent and divergent we cannot distinguish. We come from that side or that side, So convergent two proteins are the same and then they diverge. Convergent two proteins have nothing to do with each other and suddenly they get the same structure. But if you're so close to random in the actual distribution, you cannot quite distinguish one from the other. Typically people said, whenever the number is high here, I call it divergent evolution. Whenever the number is low, it is convergent. But if the vast amount of pairs is here, then you can really not distinguish between these two. Which in fact, I believe, implies that convergent evolution, in the sense of convergent to the same structure, is a major event, a very likely event. But it also means, and that is what I really made this for, the midnight zone is really something where we want to get. Most pairs of proteins that have similar structures are here. And at best we can reach this with profile comparisons.